Thank you. Thank you for your nice introduction. I'm great honor to be here to present our research journey. Recently, we discovered how the gene regulate the diabetic coronary vascular dysfunction. And as we know, like heart failure and diabetes, they link to each other with strong associations. And diabetes is an independent risk factor to lead to the diabetic cardiomyopathy. And there are so many researchers trying to identify key molecules can either block or can make it slow process to develop to the diabetic cardiomyopathy and then hinder the process to the heart failures. And however, there are so many unknown key families during this process, and after so many years, researchers and they found that JPCR family is very good and strong target and to modulate and then can make this process slowing down. And one of the major JPCR families we identified can be modified by the GRK families. And if the JPCR family cannot exit their function very well, and they either be silenced or downregulated, or they'll be post-modified, and then this heart or this response to the protective signals will be inhibited. And what the reason behind of those JPCR families during their lost their protective mechanism in the diabetes, in the diabetic cardiomyopathy, and GRK families is one of the key player. And therefore, because my research work focuses on the understand the key mechanism during this diabetic ischemia, diabetic cardiomyopathy, and we has been found that GRK2 family has a main function in the cardiomyocytes. And we also identify this, how this GRK family can make these cardiomyocytes lose their protection and lost their JPCR receptors protective effects. And we found that because during the cardiac injury and GRK2 was upregulated and then modified one of similar GPCR family adiponectin receptor and then lost adiponectin's protection in the heart. And then start from here, we start to realize GRK family, this GRK2 is very important in the cardiomyocytes itself. And it can also, because this GRK2, it can, if blocked GRK2, can make slowing down of the heart failure process. But there are another different story if we trying to understand coronary artery disease. And we using the large screening results from the single cells data, we can dissect the endothelial cells population to the 5 subcategories. And we can see here this on your left, this part of data showing that endothelial cells, they have different functions to express another GRK family members. It's not GRK2 anymore. And so GRK5 has a different population isolated from the isolated endothelial cells. We can see this part is a major GRK5 expressed the highest level, and this is a new growth endothelial cells from the heart, and others still have some GRK2 expressed high levels during the diabetic faded animal models, and we isolate the endothelial cells, we can see this part of GRK5 expression level still high, but not like this new growth endothelial cells population. And then we also trying to understand what the GRK5 expressed, what their characteristics. And then we using the obesity animal model and diabetes animal models, because we think the diabetes developed from the obesity. And we can see that GRK5 expressed almost double their expressed level in the diabetes models. And therefore, I think like in the endocellular cells, GRK5 is a key factor, maybe play some role during this diabetic coronary artery disease. And then this one could be the cause to lead to the severe heart failures. And then to identify GRK5's function, and we asked how this GRK2 what their function role, what their make the endocellular cells lost protection from the adiponectins, because we know that adiponectin can through their receptors to protect the heart. And also we want to understand GRK5 if it could be served as a therapy target during this diabetes coronary artery disease. And then we investigate what the GRK5 doing in high-fat diet-induced diabetic animal models. And we found that start from the high-fat diet, 3 weeks GRK2 was obviously markedly increased of their expressed level. And also reflect from the human samples, we can see like diabetic-induced heart failure, they are increased of GRK5. So we were thinking like validated by the human data and animal models, we believe GRK2 may be a good point we can start, continue our research. And then we isolated the endocellular cells and giving the high glucose, high lipids treatment, and we see trying to observe this GRK5's expressed tendency, and we found this even start from the 12 hours, they still can increase their expressed level. And also the but however, when we found, because we want to see if GRK5 can modify the receptors and affect adiponectin's protective function, we measure adiponectin receptor. Maybe if not GRK5 increase, it's the GRK adiponectin receptors dysfunction and make this protective role lost. And then we found that adiponectin receptor 1 levels doesn't change that much. And which lead us to understand is the GRK2, GRK5 is not the adiponectin itself has problem in the early of the diabetic cardiomyopathy. And then to focus on the GRK5's function, and we first we using the overexpressed and knockdown animal models. We using GRK5 knockdown animal model, and then to see if giving the high-fat diet-induced animal diabetes model and give some for the stress of ischemia heart, and we see the coronary vascular dysfunction, and we can see if after giving the diabetes, following with the MIR model, we can see here during the GRK5 knockout models, even gave the high-fat diet, they are have not like even worse the functions. That means GRK5 is a bad guy. If we remove it off, it didn't show that much worsening of the endocellular cells function. Therefore, we for the confer is coronary artery. It doesn't change that much if we could protect the diabetes heart cardiac function. And we still use GRK5 knockout animal models and giving the MIR stress, and with diabetes we can see that GRK5 knockout don't worse too much about compared to the MIR itself. So to confer that GRK5 is a regulator to help of can help the diabetes cardiac function, and we can see that this is a bar graph to analysis of last slide's data. We can see ejection fraction and their contracted levels can also maintain reserved from after GRK5 knockout. And because the GRK5 endothelial cells in the coronary artery, they can affect a lot of cardiac relaxation function. We're also using the EA ratio and to measure how this GRK5 can affect the heart relaxation. Of course, in the heart failures, contraction function definitely be affected that much, but relaxation function also cannot be ignored. And we see, like from the EA ratio, we can see in the loss of GRK5, it can partly recover of this relaxation function. Looks like we can see from their recovery levels, looks like GRK5 have more help in their relaxation function than the contraction function. Of course, these are GRK2 overexpressed. GRK2 is expressed in the cardiomyocytes, which is more responsible for the contraction. So these are endothelial cells of the coronary artery. If GRK5 pay responsibility to the relaxation level. And what GRK5 do in diabetic heart, what they are exactly doing in the endothelial cells. After we knock down, remove the GRK5, it shows they can recover the relaxation function. But what exactly GRK5 can regulate? And we measured all the endothelial cells, migration and invasion capacity. And we can see GRK5, of course, it can regulate endothelial cells, the migration and the invasion abilities. We're using the adenovirus to overexpress GRK5. And at the same time, we use adenovirus bring over siRNA to knock down the GRK5 using overexpression and knockdown approaches. And we double confirm that GRK5 can affect the endothelial cells migration and invasion ability. So GRK5 can affect the biologic function of the endothelial cells. And then we're trying to see in the how it would regulate the adiponectin signalings because it protects the adiponectin signalings. But GRK5 overexpressed will be bad. And then we can see that GRK5, we're giving the high glucose, high lipid models, it can modify, it can increase adiponectin receptor phosphorylation. And these are similar, of course, GRK2 and GRK5 is same family. We expected what they would do. And however, we found is they do a similar thing, like GRK5 can be phosphorylated and post-modified and to help to worse the adiponectin protection. How to make this worse? We measure the downstream signalings of like energy signal of AMPK, and we measure the ENAS. This is a relaxation function, and the GRK1 and 2. This reflects endothelial cells growing function. We found that GRK5 influence those signalings to seems to regulate endothelial cells. So many aspects in their biology, and then this we doing this from the isolated animal models of the cardiac macro endothelial cells, and at the same time we measure the human cardiac macro endothelial cells function and their signaling of the GRK5 might modulate it. And then to better understand, they affect so many signalings, how they influence adiponectin receptor phosphorylated, and how it influence adiponectin protective role. And we giving our express GRK5, we measure the adiponectin receptor. We found after 3 days, it started to dropping of the adiponectin receptor levels. That means after a receptor was phosphorylated by the GRK5, it starts to dropping their level, expressed level. This make adiponectin resistance. They cannot protect the endothelial cell anymore. So to figure out what how it would be like regulate adiponectin receptor levels, and we giving the treatment after 7 days, and we can see using the blockers trying to see who plays a role during this dropping the adiponectin receptors. We identify is lysosome degraded of this adiponectin receptor 1 after 3 days treated by the high lipid high glucose. That means in the diabetes situation, this post-modulation and can active lysosomes response and make them go up to eat all the adiponectin receptor 1 related signals. And further concluded, I using this paragraph. I use this picture to give a wrap up what we found during this research journey. And we know like adiponectin can protect the heart from the cardiomyocytes and cardiac macroendothelial cells. And however, if in the diabetes situation, they found that GRK5 was overexpressed, and then they giving the phosphorylated adiponectin receptor 1, which can block the adiponectin endocellular cells, adiponectin activated AMPK and ENAS and even ERK signalings, and then activate the lysosome degradation and make the adiponectin could not protect the endocellular cells anymore. So which lead to diabetic cardiomyopathy and eventually affect their relaxation function in the heart and lead to the heart failures at the end. So our story also to see like GRK5 is a key player in worsening of the diabetic cardiomyopathy and during this process. Apparently its major function is influence the relaxation function. So I'm happy to answer any questions and thank you for your attention.